Imagine looking at the medical records of, I think it was 2.6 million cancer patients.
Yeah, 2.6 million. It's a massive meta-analysis.
Right, a massive data set. And finding this hidden, undeniable thread, like patients suffering from depression or anxiety actually die at significantly higher rates.
Which is something medicine has recognized for decades, honestly.
Exactly. But for decades, we've kind of just, I don't know, waved our hands at it.
Yeah, blamed it on a vaguely suppressed immune system.
Right, or just, general systemic inflammation. But today, we are going to completely shatter that vague assumption.
We really are.
Because we're doing a deep dive into a groundbreaking 2025 Nature Cancer paper, and it proves something entirely different and, frankly, kind of terrifying.
It is wild.
The brain's emotional center doesn't just passively influence your immune system. It literally builds a physical wire straight into a peripheral tumor to feed it.
And what makes this research so paradigm-shifting is the sheer specificity of it. We are finally moving away from the idea of hormones as sort of floating aimlessly in the bloodstream.
Right, it's not just a hormone bath.
Exactly. This paper maps a direct physical neural circuit, and it originates in a subcortical brain region called the lateral septum, or the LS.
A lateral septum.
Yeah, and the lateral septum is, well, it's essentially the brain's switchboard for regulating mood, fear, reward, and crucially, anxiety.
Wow.
And what these researchers discovered is that colorectal cancer cells actively hijack this exact emotional center to drive their own aggressive expansion.
That is just, it's mind-blowing. So just to anchor where we are before we really get into the deep anatomy here.
Good idea.
Epidemiological data definitively links stress and anxiety to worse cancer outcomes. That's our starting point. And our mission for this deep dive is to uncover exactly how the lateral septum of your brain's anxiety center hardwires itself into your gut.
Literally hardwires.
Right. So I want you to think about this for a second. Have you ever had a strong, undeniable gut feeling when you were anxious? Like a literal knot in your stomach?
Oh, yeah. Everyone knows that feeling.
Keep that very real physical sensation in your mind as we go through this, because we are about to map the exact biological highway connecting your mood to the tissues in your colon.
And to map that highway, we actually have to start at the destination. We have to start at the tumor microenvironment itself.
Right, down in the gut.
Exactly. The researchers noted that colorectal cancer patients who have a higher density of nerve fibers, specifically nerve fibers physically infiltrating their tumors, well, they have significantly poorer overall survival rates.
So the nerves are actually inside the tumor.
Yes. The tumor isn't just incidentally growing next to some nerves. It is actively constructing its own communication infrastructure.
It's acting almost like a parasite. But, how does a clump of mutated epithelial cells actually pull a nerve into itself?
By laying down a chemical gradient. The tumor cells secrete large amounts of something called nerve growth factor, or NGF.
Nerve growth factor, okay.
You can think of NGF as a highly potent biological fertilizer, but it also acts as a directional beacon.
Like a lure.
Exactly. As the tumor pumps this out, nearby nerve endings in the gut tissue sense the gradient, and they physically grow toward the source. They eventually weave themselves right into the tumor bed.
So the tumor essentially baits the local gut nervous system into plugging in. But how did the scientists trace that local connection all the way back up to the brain's anxiety center?
That's the tricky part.
Right, because you can't exactly just put a, radioactive dye in the gut and hope it perfectly illuminates a single microscopic pathway up to the brain. Is it like dropping a microscopic GPS tracker into the tumor and watching it travel backward up the nerve highways?
That is exactly what it's like. But you need a tracker that naturally travels along neural pathways. You need a viral GPS tracker.
Oh,
yeah. The researchers used modified versions of the pseudorbes virus, PRV, and the herpes simplex virus, HSV.
Wait, rabies and herpes? Because of how they infect the nervous system.
Precisely. These viruses have evolved an incredibly specific mechanism over millions of years. They hijack the cellular transport machinery of neurons to move across the synaptic gap.
Oh, wow.
So by injecting these viral tracers directly into the colorectal tumors of mice and tagging them with fluorescent proteins, the researchers could literally watch the virus travel.
So they glow.
Right. And the modified virus travels retrogradely, meaning it moves backward from the nerve ending, up the axon, across the synapse, and into the previous neuron.
So they drop the glowing viral tracker into the tumor and watch it walk backward up the biological wire.
Step by step.
And what they found wasn't just some random tangled web, right? It was a highly specific 4-stop polysynaptic circuit. Let's trace it, starting from the top down.
Okay, at the very top, step 1, we have the lateral septum in the brain, specifically a cluster of neurons there that produce GABA. Now, GABA is traditionally an inhibitory neurotransmitter.
Right, GABA calms things down.
Usually, yes. And these specific neurons send their axons down into the midbrain to step 2, the lateral hypothalamus, or LH.
Wait, so if the lateral septum is releasing GABA onto the lateral hypothalamus, it's sending an inhibitory signal, right?
Right.
It's effectively applying the brakes to the hypothalamus.
Exactly. It's an inhibitory projection. Then, from the lateral hypothalamus, the signal travels a massive distance down the spinal cord to step 3, the sacral parasympathetic nucleus, or SPN.
The SPN, okay.
You can picture the SPN as a major neural relay station sitting right in your lower spine.
And from that spinal relay station, the nerves branch out to the final destination. Step 4.
Step 4 is the enteric neurons, the ENs. These are the intricate networks of cholinergic nerves embedded directly in the tissue of your gut.
And these are the exact nerves the tumor lured in with the nerve growth factor.
Exactly. Yeah. The loop is closed.
Okay, let's do a quick recap of what we've just built here, because it's a lot.
It is a lot of anatomy.
So tumors use nerve growth factor to physically pull gut nerves into their tissue. And using these viral GPS trackers, scientists proved a literal 4-stop highway.
Right.
From the lateral septum down to the lateral hypothalamus, through the spinal relay station, and straight into the colon. So, listener, I'll pose this question to you. If a tumor goes through the immense biological effort to wire itself into your nervous system, what kind of signals do you think it is trying to listen to?
Right. And to answer that, the researchers had to do more than just map the wire. They had to send signals down it.
How do you even do that?
Using 2 techniques called optogenetics and chemogenetics. They genetically engineered the neurons in the lateral septum of these mice so they could be manually turned on or off.
Oh, wow.
Yeah. With chemogenetics, they used a chemical called CNO. They inserted designer receptors into the brain cells that only respond to that specific synthetic drug.
So they essentially built a biochemical remote control.
That's a great way to put it.
They inject the drug, and absolutely nothing in the mouse's body reacts, except for those specific stress neurons in the lateral septum.
Exactly. They isolate the circuit completely. And the results were unambiguous. When they used the remote control to activate the lateral septum GABAergic neurons, the colon tumors grew massively.
Massive growth.
Yeah.
Just from turning on a brain cell.
Yep. And when they inhibited those same brain cells, the tumor shrank. The brain's emotional center was directly driving the proliferation of the cancer.
But wait, this is where we need to look at the cellular mechanism, because there is a huge physiological bait and switch happening here.
A huge one.
We established that the gut nerves at the very end of this circuit are cholinergic, meaning under normal circumstances, they release acetylcholine to help your gut digest food.
Right. Rest and digest.
So why does a normal digestive signal suddenly make a tumor grow exponentially?
That is the crucial pivot of this entire paper. When that lateral septum pathway is highly activated by stress, it physically alters the behavior of those gut nerves.
It changes what they do.
It changes what they release. Instead of releasing their normal acetylcholine, the circuit forces those enteric nerves to release a massive, localized dump of GABA directly into the tumor microenvironment.
Wait, GABA, but that seems completely counterintuitive. Up in the brain, like we said, GABA is the great calmer.
Yes.
It opens chloride channels to inhibit neurons and slow things down. Why is GABA acting as a potent growth fertilizer down in the gut?
Because the tumor cells are not neurons. And in biology, a signal is entirely dependent on the receptor that catches it.
Okay.
The cancer cells have evolved a highly specific mutant receptor to capture this GABA.
I want to give you, the listener, an image to hold on to here to remember this. Think of the tumor cell like a radio.
Good analogy.
Normally, a rogue radio shouldn't be able to pick up encrypted private neural broadcasts. But the tumor has mutated to build a highly specific, unauthorized antenna, just totally rogue piece of hardware. In the paper, this is the GABA-A receptor containing an epsilon subunit. Let's just call it the epsilon receptor. Or, picture it as an evil ear, shaped like the Greek letter epsilon.
That unauthorized epsilon antenna analogy is spot on. The tumor heavily overexpresses this GABA receptor. And when the gut nerve dumps GABA into the surrounding tissue, it binds perfectly to this epsilon antenna on the cancer cell.
But it doesn't calm it down.
No, Because it's a cancer cell and not a neuron, the result isn't inhibition, it's catastrophic depolarization.
Let's drill down into that mechanism exhaustively. What exactly is happening inside the cancer cell when the GABA hits that evil ear antenna?
Okay, so when the receptor binds GABA, it alters the electrical charge across the tumor cell's membrane. That's the depolarization. This voltage shift forces open specific calcium channels.
Calcium channels open up.
Right. Suddenly, calcium ions, K2 plus Ac-ura, rush from the outside of the cell to the inside. Now, in cellular biology, a sudden flood of intracellular calcium acts as a massive alarm bell. It's a powerful secondary messenger.
And that calcium floods into the nucleus to change the gene expression.
Precisely. Yeah. The calcium influx triggers a signaling cascade that specifically upregulates a protein called TSPAN1.
TSPAN1?
Yeah, and TSPAN1 is well known in oncology. It is a membrane protein that fundamentally alters the cytoskeleton of the cell. It drives aggressive cell division, and it helps the cancer cell anchor to and degrade the surrounding extracellular matrix.
Which is how it spreads.
Exactly. It's the primary driver of invasion and metastasis.
So let's connect the whole chain. The brain is stressed, the lateral septum fires, the signal travels down the spine to the gut nerve dumps GABA instead of acetylcholine.
Yep.
The tumor's unauthorized epsilon antenna catches it. Calcium floods the cell, TSPAN1 levels spike, and the tumor aggressively multiplies and invades.
It's an unbroken chain of causality.
It's terrifying, but the researchers proved they could break the chain, right? And they did it using something you'd literally find in a dermatology clinic. Botox.
Yes, botulinum toxin A, or BTXA.
Botox.
Botox is one of the most potent neurotoxins we know of. It works by cleaving the snare proteins inside a nerve terminal.
And without scare proteins.
Without them, the little vesicles carrying the neurotransmitters cannot fuse with the nerve membrane. They are physically trapped inside the nerve.
So when the researchers injected Botox directly into the gut tumor, it chemically snipped the wire.
Exactly. They paralyzed the nerve endings. Even when the brain's stress center was firing on all cylinders, sending signals down the spinal cord, the enteric nerves were physically incapable of releasing that GABA into the tumor bed.
And the result?
The stress-induced tumor growth was completely halted.
That is incredible. Okay, let's do another quick checkpoint to lock in these mechanisms for everyone. Activating the brain's lateral septum forces gut nerves to perform a biochemical bait and switch, right? Dumping GABA onto the tumor. The tumor uses that unauthorized epsilon antenna to catch the GABA, which floods the cell with calcium and boosts the invasive protein, TSPAN1. And critically, injecting Botox locally paralyzes the nerve and stops the growth in its tracks.
You got it.
So consider this, listener. If Botox can sever the communication between a tumor and the nervous system, could localized nerve blocking become a standard pillar of cancer therapy alongside chemo and radiation?
It's a profound clinical question. But to fully grasp the clinical reality, we have to move away from genetically engineered mice with laser remote-controlled brains.
Right, because humans don't have optogenetic brain implants. How does a mammal naturally flip this circuit into overdrive without a synthetic drug?
Through severe chronic psychological distress. To prove this, the researchers used a model called chronic restraint stress, or CRS.
What does that involve?
For 2 weeks, mice were placed into tightly confined spaces for several hours a day. It doesn't cause any physical pain or tissue damage, but it induces immense psychological panic and anxiety.
It's like severe claustrophobia. And after 2 weeks of that daily stress, they looked at the chemistry in the gut. What actually happened at the biological level to flip those nerves from acetylcholine to GABA?
It comes down to systemic endocrinology. The chronic psychological stress caused sustained elevated levels of corticosterone.
Corticosterone. That's the mouse equivalent of cortisol, right? The human stress hormone.
Exactly. This stress hormone constantly bathed the enteric nervous system. Over 2 weeks, that corticosterone physically altered the gene transcription inside the gut neurons.
It changed their programming.
Yes. It downregulated the enzymes responsible for synthesizing normal acetylcholine and heavily upregulated the enzymes that manufacture GABA.
So the chronic stress hormone literally rewired the manufacturing plant inside the gut nerves, turning them into GABA-producing factories.
And as a result, the growth of their colon tumors just exploded.
But mice are mice. The real question, the one everyone listening is probably asking, is whether this physical, measurable footprint of psychological stress exists in human oncology wards.
And this paper provides highly compelling evidence that it does. The researchers analyzed advanced 18 FFDG PET-CT scans from 103 healthy individuals and 103 human patients diagnosed with colorectal cancer.
Wait, they weren't looking at the tumors in these scans, were they? They were looking at the human brain.
Right. Specifically, they looked at the septal region, which is the human equivalent of the lateral septum. They measured the metabolic uptake, essentially how much energy that specific brain region was burning, measured as SUVR.
And what did they find?
The data showed that patients with colorectal cancer exhibited significantly higher metabolic activity in their septal region compared to healthy controls.
Their emotional stress centers were running incredibly hot.
Exactly.
But the truly staggering finding wasn't just that cancer patients are stressed. I mean, we know getting a cancer diagnosis is incredibly anxiety-inducing.
Of course.
The breakthrough was the linear correlation they found.
Exactly. Among those 103 cancer patients, the individuals who had the most metabolically hyperactive septal regions in their brains also had the largest primary tumors in their colons.
Wow.
The correlation between the brain's energy consumption and the tumor's physical volume was direct and undeniable.
So we can look at a human brain scan, see the physiological footprint of severe psychological distress lighting up, and mathematically correlate it to the size of a malignant mass in the gut.
Furthermore, we can identify which human patients are most at risk of this phenomenon. Colorectal tumors are classified into different consensus molecular subtypes, or CMS.
PMS.
These subtypes dictate the genetic landscape of the tumor. The researchers analyzed human transcriptomic data and found that patients with the CMS2 and CMS4 subtypes naturally express much higher levels of that epsilon receptor.
So if you have a CMS2 or CMS4 tumor, your cancer has essentially built thousands more of those unauthorized epsilon antennas.
Correct. And the clinical outcomes absolutely reflect that vulnerability. Patients with those specific subtypes, who also showed high epsilon receptor expression, had significantly worse overall survival rates. They are uniquely susceptible to the stress signals their own brain is sending.
Okay, let's bring all of this together for our final synthesis. Chronic, real-world psychological stress floods the system with hormones that chemically rewire gut nerves, turning them into GABA factories.
Right.
Human PET scans confirm that hyperactive emotional centers correlate directly with larger colon tumors, and specific tumor genetics, like the CMS2 and CMS4 subtypes, make some patients highly vulnerable to this exact brain-to-tumor neural pathway.
That sums it up perfectly.
So listener, knowing that chronic stress leaves a measurable physical footprint on a brain scan that perfectly matches tumor size, how urgently should we be treating psychological stress in oncology wards?
If we step back and look at the totality of this deep dive, it really represents a fundamental shift in our understanding of oncology.
It does.
We began with a purely observational statistic that anxious cancer patients have higher mortality. And we have drilled down through the anatomy all the way to a single epsilon receptor catching a neurotransmitter and flooding a cancer cell with calcium.
It proves that the tumor is not an isolated lump of tissue. It is a highly active, highly adaptive participant in the body's ecosystem, literally growing its own wires to plug into the host's emotional state.
The bidirectional communication between the mind and the physical pathology is no longer a philosophical concept. It is grounded in hard anatomical science and molecular biology.
So what does this actually mean for you? For decades, the standard of care in oncology has been to attack the tumor locally. We use surgical excision, blunt forward chemical poisons, and targeted radiation to kill the cells in the colon.
Right.
But if a tumor heavily relies on a continuous, hardwired neurological phone call from your brain's stress center to maintain its aggressive growth, does our entire approach need to evolve?
It might have to.
Could we see a future where aggressive psychiatric intervention, targeted anti-anxiety medications, or even local neural blocking agents like Botox aren't just considered palliative care to make someone comfortable? What if treating the mind becomes a prescribed primary biological prerequisite for curing the body?
It forces us to view the patient as an integrated neural system rather than just a host for a localized disease.
That diagnostic X-ray we mentioned at the very beginning, the one that feels so perfectly clear when you're looking at a broken arm, but historically felt so murky when trying to understand the mind-body connection. It turns out the image wasn't murky at all. The biological wires connecting our anxiety to our physical illness were always there. We just didn't have the right lens to see them. Until next time.